That's Steeve from Duncan. Steve's the youngest member of the group. He's been doing this maybe only 12 or 13 years. And still one of the most effective PhD doctors. You can see there's one of those unique people, like Duncan, who looks after patients, but also does research in science on tissues, cells, and animals. And to be one of the breed that we have that we're very proud of, clinician scientists, for helping us understand and making things a lot better. But then my dear friend Dylan laughed. When we walked around and had lots of people, a lot of people seem to have you as a doctor. What's that?
My condolences.
And they said to me, oh, he's wonderful, he's so smart, and I'm their guy. PhD doctor and also ICU doctor, and it's been an honor and pleasure working alongside you for 20 years. So thank you very much, all three of you, for joining us. So in terms of medications we have, all of them fall into the pathways that we talked about earlier today. On the left side, too much endothelium that not able to prescribe, and we have endothelial antagonists, as you know. Then also we're very early on showing that patients with pulmonary hypertension need less nitric oxide. We have medications that have nitric oxide work better. On the last slide, prostacyclin, we don't have enough, and we have ways of getting it back. So in terms of the newest drugs, we have all of those three pathways. So first I want to talk about briefly is macitentan or oxalate, one of the endothelial receptor antagonists. And so it is out there, for example, in Quebec, luckily it is funded by the government, so lots of people are on it. In the rest of the country, there might be, I think I heard the number, somewhere around 400. It's not a lot across the country, and a lot of that is compassionate release from the company who makes it. So we have not been able to get funding anywhere outside of Quebec. So maybe I'll just turn it over to John. John, just a couple of ideas about the use of oxalate in pulmonary hypertension.
So thanks, Sanjay. Well, it's the new kid on the block as far as the endothelin receptor antagonist, which I'm sure you talked about, blocking that important vasoconstrictor. And you mentioned Duncan Stewart had done a lot of the seminal and early research and the importance of that compound, not in constricting blood vessels, but also altering the way they grow and respond. And so it's always been viewed as an important some substance in the body which contributes to the disease, and so blocking its effects has always been something that people have been looking towards. It's one of the three pathways that Sanjay mentioned. It's kind of fourth in line really, and they had bosentan, cytaxanten, which was pulled from the market, ambrisentan, and now macitentan. Macitentan as a compound is perhaps in theory more effective. It penetrates the tissue more effectively. It's once a day. It has perhaps fewer side effects compared to some of the earlier compounds, so patients may stay on drug a little bit better. They don't have as many liver side effects. They tend not to get the swelling in the legs that you sometimes would get with some of the other medications once in a while. And it also has less interaction with other medications, so people who are on some diabetes drugs or some anti-rejection drugs can actually take this one as well without it messing up your other medication, and also less interference with some of the other pulmonary hypertension medications. And it's the first drug really that, the earlier trials where we looked at the effect of these therapies on you folks with the condition. We looked at how far you walked, what your pressures were in your lungs and other things. So we didn't really look at, well, what's the long-term benefit beyond just sort of 12 weeks of treatment? Like, does it keep you out of hospital? Does it keep you away from transplant? Does it keep you alive? Does it keep you happy? And the oxalate or macitentan was actually tested and shown to actually contribute to keeping people out of hospital, preventing them to have to go on more advanced treatments like intravenous treatments or treatments, injection under the skin with prostanoids. So to that end, we think it as a compound, it's a better drug. And then when we put it to the acid test, which was the perhaps more sophisticated trial answering some of the really important questions, it was shown to be beneficial. So as a result, many of us view it as far as the endothelin antagonist group, perhaps the more desirable drug. But one size doesn't fit all. Some people don't tolerate it, and they tolerate the older compounds. So what works for many doesn't necessarily work for you as an individual patient. So it's just on average averages, right? So we try different things, and although trials look at groups of people, when it comes down to the individual, you have to trial and error very often.
So if it's available, some people are on it, not going to get done well, doesn't mean everybody should be on it, and we are negotiating with provincial governments about funding. Let's move on. So the second half is that nitric oxide pathway we talked about. And we have so that filter body, like this is out of that many of you are on, I know. And we have this new one, real Sigma. Now, real Sigma is unique, as you said. It also works with patients with chronic thromboembolic CTAD. It's the only drug approved for that. And many patients are on for that. It is not funded, unfortunately, for PAH, so it's not a challenge to use. Maybe I'll ask Steve, any thoughts about real Sigma use in PAH patients?
I think it's the first Latin trial that proved that real Sigma was effective. It was like the first trial in that pathway showing it on top of baseline therapy as a second line therapy. Usually we had some concerns about side effects of that drug in terms of hypertension, but at the end of the day, it turned out patients do have quite limited side effects with that drug. It is to be taken 3 times daily, however, so it's my bothersome some people to take this, and certainly for the CTAP patients, this is the first time that we observed a drug that was effective for those patients. So this is a major advantage for those patients.
There is some case that if you're on one drug in that pathway, it's a medical downfill, and it doesn't work, could you take a new Sigma? I'll ask the panel of results, any ideas on that?
For us, of course, patients that are already in sildenafil or tadalafil should not take real Sigma at the same time. Of course, we start to switch from sildenafil to tadalafil, switching those patients to real Sigma. There's been some publications recently showing that with some very selected patients that there might be some data switching, but those who did not respond to sildenafil to tadalafil. Testing was not perfectly done though. It was what we call an open label study, so it was not randomized. But there is an ongoing trial that will try to answer that question, whether patients who are failed to sildenafil or tadalafil should be switched to from using and we are waiting for those answers.
So we don't have, as Steve mentioned, we don't have robust data to address the question, but from a sort of theoretical point of view, there's reason to think that there could be benefit because the phosphodiesterase inhibitors like tadalafil only reduce the breakdown of the mediator, so and they're dependent on the endogenous nitric oxide that's being produced. So we know that in patients with pulmonary hypertension, the endothelium, the inner lining of the blood vessel isn't working well, and very often it's not producing enough nitric oxide or not in the right place. So the advantage of real Sigma is that it actually directly can stimulate the enzyme, independent really of having nitric oxide, a mediator there. So theoretically, it could increase the activity of that pathway in a place where there's insufficient nitric oxide to really make it work.
That's important. So it could work in some people, even if you don't make nitric oxide. But we see that very carefully, not everyone needs a solution for doing well, and some people do not respond to sildenafil tadalafil as a possible option that may work for the scientific reasons like we mentioned. Now, in that same pathway, if you're not making nitric oxide anymore, what about just breathing it in? It's a gas. And in fact, if I look around the room, hands up, somebody here that's walking around with nitric oxide. Great. And so this is new and exciting. Nitric oxide is a gas that has been used in the hospital, in the ICU, and other places for at least 20 years. A long, long time ago, when I was doing research with Duncan and others on the field, we had tried nitric oxide in different things. And so the idea has always been, can we bottle it like oxygen and can send it home with it? If you can then deliver to yourself reliably, minute after minute, for 24 hours a day. And we, for a long time, did not think that was doable. But it's something we, I am Therapies, now Icaria, has come up with what's called a mini cylinder. I think it weighs 5 pounds. You want to show us the cylinder? There it is. It's actually got finished down to 2.5 pounds. So that is supposed to give enough nitric oxide for 24 hours. And so tomorrow they just switches it out for a new cylinder. So it's quite an interesting idea. We've done a couple of studies, not the most promising in terms of a large effect, but a small effect in certain people. So we are doing another study, which is quite promising. I don't think it'll be a treatment for people by itself, but adding it on to something else that seems to make sense. It's something that you need to hear more about as another therapy, nitric oxide. A bit of a challenge. If you're wearing oxygen, you already have something in your nose. When you add nitric oxide, they actually have a way to do both of them together. And that actually seems like it works quite well for running on oxygen. So you will hear more about that. Let's focus on the last factor. If you remember this morning I mentioned prostacyclin, most of us believe is probably the best treatment we have. If you had bad pulmonary hypertension, if I had bad pulmonary hypertension, I would like to see on prostacyclin infusion tomorrow because people respond the best and they have the best response now and in the long-term future. And yet, it's not easy. I have to look at the trumpet and cassettes and program that keep my mind sterile. I like to deal with the side effects in the skin. So we use it, but not as much as we might be able to benefit. But it is important. Not everybody's the same. We heard the story of Judith at lunchtime, and she unfortunately had a lot of trouble with subcutaneous infusion, terrible pain. That's not universal. Lots of people tolerate it well, and they've done very well, including patients that I looked after. The intravenous has been a godsend since we had it 20 years ago, and some people would rather not have a victim catheter sticking out their neck because they get infections on them. So all of them have people that benefit a lot, and they all have side effects that limit the benefit in some people as well. We don't use them as much as we could. Now, maybe I'll ask something. Let's work with a company of the U.S. called United Therapies. It's quite unique. They have developed prostanoids for intravenous, subcutaneous skin, a pill, and inhale. Every single way you think of getting it. Maybe not everywhere, but I don't want to think of everywhere. So, Duncan, any thoughts about the inhaled prostanoids? And we haven't really ever had it in Canada. Have you heard something from your partners in the U.S.?
And so I think obviously the ability to give it directly to the lungs with inhalation has major advantages because you're not going to get side effects elsewhere in the GI tract and headaches and other things which are common with the prostanoids. They're very effective, but they don't have unwanted effects if they get when you increase the levels elsewhere. I think the experience has been that it's hard to get the same degree of clinical benefit from the inhalation. So I think there's some benefit, but it is clearly more modest, less robust than the intravenous or perhaps even the sub-Q delivery. So that's why it's not available in Canada, I think, because it's been hard to convince the regulators that there's sufficient benefit to actually provide it to Canadians. But I think certainly the experience of colleagues in other countries has been that there are patients who benefit from this, and it has a niche, and the convenience of being able to deliver directly along the leg or minimization of other side effects is very helpful. So again, everybody is different. I think all of us that everyone responds differently. That's why it's good to have a range of different therapies and find out which one works for an individual.
Wonderful. There is an oral form as well. You can see it there, Provencher, so it is the same as prostanoid that you use subcutaneous, so a little modification, and it is available in the U.S. It's quite an expensive medication, not available in Canada. John, have you guys been part of the studies with oral modulins or anything?
Yeah, we hope they're rare. We are, or were, and it continues on. It's the trial that never stops. It's ongoing, and I think again there may be some patients who benefit from it, but it's a difficult pill to swallow. It has a lot of side effects, and particular intestinal side effects, diarrhea, stomach upset, same sort of stuff you would get by the intravenous route. So I think the trial wasn't breathtaking in its results, so I'm not sure of its exact role in the current landscape. So I must say I don't lie awake at night worrying that I was necessarily missing it, particularly with the other compounds potentially coming.
Yeah, that's probably my sense as well. We don't have the inhaled neural problems like that. Hopefully our patients are disadvantaged. The really have never been tremendously effective. They're largely add-on. If you're on already 2 medications, maybe 3, and you have this on for another little bit. I don't think it will do so much. Very different to that is the last one on the list. It's very important because in this pathway, it works on the same receptors on the surface of cells. These proteins up there looking for something to react with, and prostacyclin lands on those proteins and causes benefits. And we don't want to use prostacyclin with all the side effects. So actinium generated hormone that is not a prostanoid, that acts on the same receptor called selective hydroxyapatite. Now this is quite exciting. The largest study in cardiac hypertension ever done was published a year and a half ago. 1,100 patients showing quite strong effect by itself or on top of other medications, including the beta we were taking. It is not yet funded. It is not tentative approved, except in one place, and that is Quebec. Well, it's not Canada approved, but we. Well, also it is. I thought there was exceptional access. No.
Well, we have a special access program in Quebec, but we can have access to safety in some patients. It's not officially
and so you have to experience with people in the trial?
We get experience as part of the trials and we just started that and people as well. Well, this is very attractive, of course, because it targets this pathway. Although it is in early, we started driving increase the dose progressively because even though this is not the true prostacyclin, it has some side effects, maybe look like prostacyclin, including headaches, jabbing, and so on. We found it is generally less severe than prostacyclin or prostacyclin. And of course, it is exciting because I think it might delay or in some cases avoid the need to IV or subcutaneous prostacyclin. So this is a very interesting drug for communications. Of course, there is some fear about the fact that some patients who really need IV or subcutaneous prostacyclin would refuse that and select and want to try selectivity delay drug that would be very benefit for them. But I think there's very niche for the drug communications, likely as a second or perhaps even third line therapy for communications.
So as we said, they're having trouble hearing that psoriasis. And so I think that's really important to see. And most of us agree with that. It's very exciting, obviously, to have a molecule pill that might one day be able to do what the prostanoid infusions have done. Again, these are the best treatments we've ever had, and all of us would say if you're doing well on an infusion therapy, this is not the right idea to switch to because it's the same pathway. You need a lot more experience, first of all, starting in other patients and seeing how well it works over the long term. And it is potentially feasible that over the next few years, once we get experience with it, and then patients who might switch for other reasons, and you would mention that, she could tolerate sub-2, then switch to lovely has done very well. So that is a possibility. We obviously want to be very cautious about that right now. Any of the thoughts and panelists of any of these therapies for the 3 standard pathways before we move on? All right, so please.
You're going to talk about upfront or combination, or are you
We weren't actually talk much about sort of approaches to therapy. I'm maybe happy to take a few minutes to do that. Most of you know, standard approaches in because of funding reasons, like through allows us to use one medication, and typically 3 to 4 months later to reassess you. If we don't think you're well enough, or you don't think you're well enough, and we have criteria for that, then they're allowing us to add on a second medication. So it's what we call sequential combination drug therapy. It is something that's been standard around the world for many years, but there is an idea that if you're not going to do well with one drug, and we believe most people, one drug is probably not enough. Remember we said nobody's been cured of this disease, and so should we not be more aggressive? Why not be like a cancer or some other severe illnesses? Let's treat you more aggressively so you're better in 3 to 4 months, and you're likely to stay better longer. So maybe I'll ask John just to address that idea of treating you more aggressively at final combination.
Well, I think you alluded it to. It was interesting what you said, that if you had pulmonary hypertension, you would start intravenous Flolan. I think many of us feel that way who treat pulmonary hypertension, is that it's a lifestyle decision, and sometimes we see, right? But I must say, for the first time, I'm actually leaning. If I had pulmonary hypertension, I would probably choose upfront combination therapy to see how far I got. The rationale is exactly as you said, that you're looking at multiple pathways, you're getting aggressive upfront early on, and maybe maximizing the opportunity for a response. Again, it varies from person to person. Some people have a very remarkable response to that strategy, and other people don't. And I think it's just a matter of figuring out who that person is, but more importantly, reevaluating it and keep on pushing and trying different things, be they mainstay therapies or novel agents, which we'll be talking about later on as they come through. But it's clear that given and Duncan and Steve have contributed the science here, we recognize that there are multiple things going on, and not any one of those things going on is what's causing this disease. And so blocking as many pathways as possible that are causing those abnormalities in constriction or abnormal growth and repair is a strategy, just like cancer and just like some infections. We recognize that it takes more than one drug. Tuberculosis, it takes many drugs to treat. Cancer, you use more than one drug to treat. And this is very much like a cancer, the way the cells grow. So using more than one drug at a time makes a lot of sense.
Wonderful. All right, let's move on. So those common treatments we have kind of variably available around the country. Let's look at the new and exciting treatments, some of which are being used now as part of clinical research. And if you recognize these, some of you may have been in clinical trials at different centers across the country, and some of these haven't quite come to clinical trials, so you may not have heard of them. But these are quite exciting from our side. And one of the big reasons is we showed you the 3 pathways, and the building nitric oxide, prostacyclin. These are not molecules or chemicals that work for those pathways. They are other pathways, which is really exciting, because we believe to some degree that those 3 pathways can kind of maximize the benefit we want to get with the medications we have. And so now it's time to look elsewhere. Can we make a bigger impact on this disease? Can we make it dramatically better by understanding the science better? So we're going to start with this idea. You wouldn't be surprised to hear that in the blood vessels that feed your lungs and pulmonary arteries, besides the blockages that we talked about earlier, cells growing, there's inflammation there. And you measure that inflammation in the blood. And so people have tried medications to turn down inflammation. One of the most common ones is prednisone or cortisone. Hasn't done very much. They're probably too generic and not focused. So there's ideas about some things that might actually work to control inflammation very specifically in the pulmonary arteries. And you can see a list there. Some of these have been tested, and they've had some very good results. We want to talk about these a little bit to give us an idea. And so maybe I'll start with Duncan. Duncan, the RTKIs, and I left that to spell it out because it's long and complicated. TKIs are the general family of chemicals that we have in receptors. And in that way, the first word there was used in a few different studies up until probably 5 or 6 years ago. Duncan, were you part of that, or do you have experience talking about that? Please.
Turn this on. Yeah, our center did participate in the trial. So it's, as Sanjay says, this is a different way of approaching the disease. These agents block growth factor receptors. So basically, these RTKIs are inhibitors of pathways by which growth factors work to make cells divide and they can get larger. So the idea was this was a way to sort of prevent that thickening of the arteries and hopefully reverse it. I think that was the INCREST program. Was a well-designed, it was a really good program. Global, so centers across the world participated. And actually the results from efficacy from looking at the ability to improve pulmonary hypertension, to improve patients' ability to walk and reduce the pressures, it actually was quite positive. So there were significant improvements in 6-minute walk, which really it's rare that we often see with that therapies, and there were important reductions, very significant reductions in all of the measurable catheterizations, decreased pressures, increased cardiovascular, decreased resistance. All that was good. The problem was unanticipated serious side effects. So the main thing was subdural hematomas. So there were 8 patients that developed this out of, I can't remember how many, probably
somewhere around 80, like 10%.
Yeah, about 10% developed this, which had not been seen in cancer. These agents had developed cancer. That's what they've been developed for, and they were useful for certain kinds of cancer. And they know this experience has not happened. But what was different was, and again, when we borrow a drug from another field, you have to be mindful of this, is our patients tend to be on blood thinners. And this agent did not study in people that are on blood thinners. So probably what happened is a combination of this agent plus the blood thinner made it more likely that a hemorrhage in the brain occurred. And so that was quite devastating. And despite the promised effects, obviously the development was discontinued because that was considered unacceptable in terms of side effects. And the other, unfortunately, the other agents, but one dextroaminase, also was caused problems with the heart, so that had to be discontinued. And other of these agents actually have been implicated in causing pulmonary hypertension. Now, a number of cases of pulmonary hypertension that have been occurred in patients taking this drug for cancer. And so what we learned is these are complicated agents. They're not very specific. They block families of receptors. And depending on which families they block, they can be helpful or they can be harmful. And they do come at a price because there's obviously significant side effects.
Super, thank you. So unfortunately, an experiment that seems to have some benefit, but it's a really unpleasant side effect of bleeding in the brain, and obviously it tends to continue those. And that pathway of growth factor receptors, so growth factors make cells grow. You want to stop that. There is a lot more work going on that we hear about. Let's go down to the next slide. Benefits. It's kind of an interesting word. I don't know where that comes from. And it's something that's new just the last couple of years. This is study going on right now. We're going to be able to recognize that Are you guys see John our study? John, you want to address a little bit about your comments?
So that stems again from research looking at inflammation. It looks at a particular blocking a particular pathway, which is important. It's kind of the inflammatory response in this instance in the blood vessels, around the blood vessels in the lung, and may contribute to those, again, the abnormal growth of the blood vessels. So it's blocking that particular pathway. And that was called, it's now closed. It was the Liberty trial. It's Eiger Pharmaceuticals, a new kid on the block, as it were. And really stems from some very well thought out science. So this was not a large study. It was basically looking more at safety and looking at kind of proof of the concept that this might work, because it's a medication which interferes with the immune system and the response to infection potentially. You want to make sure that it's not going to lead to more infections or more serious infections. So they really wanted to look at that first before they then committed themselves, and importantly the patients, to a larger study if there was a safety concern. And then so once you're reasonably convinced that the drug is safe in a small group of people, then you can venture into a larger study if it looks like there might be some benefit. So they paused, they're going to look at the results and then determine whether they want to move forward.
Well, it's quite exciting with genetics. It was actually very safe in humans. That's to show you this medication is been available around the world already in certain types of chronic leukemias. So many patients are taking benefits for many years. It's a bit of a different dose of pulmonary hypertension, but we're not hopeful that it's safe and it's not effective. We will be doing more on that definitely in the next couple of years. Paradoxolone is also an interesting molecule that works on the inflammation pathway, and it's something that they've tried to target a little more selectively for patients with scleroderma and that family of pulmonary hypertension. Steve, were you involved in either families or learning trials?
Yes, so we are. I think one thing that the other way that drug works is also that it influences how the cells produce energy. And what's very interesting is that I think it is the first study that showed that the supergroup of patients with scleroderma have more benefits than patients with the idiopathic PH, which is generally the opposite. And the other thing that was strange was that patients had less shortness of breath, they were able to walk farther, but with minimal improvement in hemodynamics. And one of the ideas is that people are short of breath during exercise because of the pH, of course, also because of their muscles that are less tolerant to exercise. And one hypothesis is that drug might improve the muscles of PH patients, and this might be a way, at least partly, the way it works.
Wonderful. Yeah, it's actually quite exciting. As Steve said, we have not seen medication in the past that ever worked better for scleroderma pulmonary hypertension than other times. And so it's more an idea. All the treatments that we talked about, we kind of have used in pretty much everybody with PAH. Idiopathic, with no cause, scleroderma, and others. That doesn't make sense. We need to learn more of which drugs work best in which patients. This is a great example of that. The next one on the list is rituximab, which maybe you may have heard of. It's actually an medication that's been around for probably about 7 years, and it's used quite widely in the field of rheumatology. Patients with things like rheumatoid arthritis, or lupus, and other severe chronic inflammations. Rituximab is an infusion therapy, but short term, over an hour, so that you can do repeatedly every few weeks or every month. That's actually been one of the best therapies that we've seen in those other kinds of inflammatory diseases. And so there is an idea that maybe similarly effective in pulmonary hypertension. Duncan, do you want to mention something about rituximab?
No, I don't really have any experience with it. So I think it's really interesting, to have an opportunity to try these compounds for these drugs, which have been developed and studied well in other diseases, inflammatory diseases. We know that they're reasonably well tolerated, and it's really good science that would suggest that inflammation is very important in pulmonary hypertension. We don't really understand it, but clearly there's a lot of information there, and it makes a lot of sense that that's contributing to the progression of the disease and symptoms that folks have. So I think it's a great opportunity. So I'm excited to see what comes out of this, and hopefully we'll find some of these agents do have significant benefit, and there'll be a role for them in treatment for patients.
Fantastic. And the last one on the list, FK506, also Steeve, that's also not a new medication. It's been around for a long time. It's used fairly regularly in transplant to prevent rejection. And so there is ideas that these drugs work in other types of diseases. There are some lung diseases that we use to as well. And so John, having done a perfect transplant, any thoughts on FK506 in pulmonary hypertension?
So, again, stems from work in the States with Marlene Rabinovich, used to be up at Sick Kids and moved south, and they looked at it. It was kind of interesting. They did kind of a search, kind of a drug directory search to kind of see what drugs kind of targeted this particular pathway and in stimulating some of the things which might be deficient in people with pulmonary hypertension. They landed on this particular agent, which is a common drug for those of us who deal with transplant, this tacrolimus or Prograf. So the trouble with that drug is when it suppresses the immune system again, and when it uses high doses, it has a lot of toxic effects, particularly on the kidneys and the nervous system. But the really cool thing about this is that the doses you need for pulmonary hypertension seems to be much lower than what we would use for preventing rejection. And it's almost below detection of the blood test that we use to detect it. It's actually used so low. And so the initial stuff in animals and subsequent stuff in a couple of reports in some people with very advanced pulmonary hypertension showed a very impressive result, and that led the investigators to do again kind of a safety and proof of concept and dosing study to determine does it actually work, and is it safe at the doses in this particular group of people. And so that's kind of in the ongoing phase, and they're kind of reevaluating again and going on. And I must say it's tantalizing enough that we've actually, I must say, personally used it in some of our patients and the only people we've ever exposed them to the risk is that somebody who was going to be on that drug anyways. So for people who once in a while, if they're on the transplant list and they're going to be on this drug anyways, we've kind of said to ourselves, well, we're going to be using a lower dose than you're eventually going to get, so why don't you try it now? And when you're at the end of the armamentarium and you're waiting for a transplant, there doesn't seem to be a big downside in trying something you're eventually going to be taking anyway. So we have done it, and I can't really comment that I've seen the same sort of response that they saw in the States. We've never really taken anybody off the transplant list after going on that treatment, which wasn't the case in those cases in the patients that they had the initial experience on.
No, I was just going to ask John what the result was. Whether there was any obvious signal.
There was no religious experience.
No religious.
No conversions.
No conversion.
All right, let's move on. This is something I mentioned a couple of times, and we're going to really show you some pictures. So your blood vessels are not wide open like normal in pulmonary hypertension. They get plugged out in many things, and one of which is your own cells are growing. Muscle cells are growing around the blood vessel, but also the cells that line the inside called endothelial cells. And so this is something that Duncan has been working on for much of his career, and Steeve is also working on this recently. And so these cells seem to grow too fast and also reproduce and multiply too much. And so clearly trying to turn that off sounds like a good idea. It's not easy, because if you want to turn that off, you want to turn off some normal cells that have to grow to keep you alive and healthy. So it's quite a challenge. And so there are some ideas. So mTOR is the pathway, again, as I mentioned, kind of responsive to the growth factor, making cells grow. And rapamycin is a treatment that works in that pathway to slow down cells growing. Rapamycin also is not a new medication. It's been used for a long time, used for some serious lung diseases and kidney involvement and some rare diseases. And so I don't know if you want to tell us about mTOR and rapamycin indication a little bit.
So I think, again, it's a really exciting idea. And as you mentioned, there's been some success in another rare disease, Granton. So a very intriguing, difficult disease that is like cancer, and like pulmonary hypertension, too much growth of some cells. So I think it'd be very interesting to see what the results will be. Again, it's a drug we know quite well. We have a pretty good idea of its tolerability and its safety profile. So again, it's very important. Again, this is one of the critical things for us is to do design clinical trials so that we can really assess whether something is working well or not, well taught or not. So it's very important so that if it isn't working, we don't use it. And if it is working, then we make sure we provide it to people that with benefit. So we'll wait and see.
Fantastic. The next one, DCA, hydrochlorothiazide, is really interesting. It's not a drug, it's kind of a common chemical that you both hear and buy. And it is something that seems to be quite exciting for pulmonary hypertension because as cells are growing too much or too fast, obviously you need lots of energy. And so it's wonderful work that came out of Alberta, colleague of Duncan, and also colleague of Steeve, Duncan, Sebastian Rene, or Donat in Quebec, on the cells changing how they get energy. And that's not a healthy thing. It's almost like a cancer cell to so hydrochlorothiazide seems to try to change that metabolism back to normal of how the cells use energy. Duncan has done a lot of work with cells and animals to show that it's quite effective. Hasn't been able to do this in people, but there's a small study of 20 people that seemed interesting. I have not heard data about that. We finished the study, but the data is coming.
Yeah, paper is going to be published shortly, and essentially what's interesting about that study is that DC was effective to treat patients, but what was even more interesting is that part of the patients did respond very well, in part in that respond, and there was an explanation, they could go back and look at the blood samples that were taken earlier in the trial and identify why some patients did respond and some patients did not respond. So it is probably one of the first steps of what we call personalized scheme, but which means to try to identify right at the beginning which patient is more likely to respond to the drug as compared to the other patient. So I think in that aspect this is very interesting. And well, again, this is a known drug. It's associated with very limited or no side effects, and the doses are used and engaged. So hopefully, someday we might be able to target patients that are more likely to respond to that.
Very exciting. This is an idea that has been out there for at least 20 years. I've been doing cardiac imaging, responders and non-responders. Again, there's no guarantee if we give you medication X, the quality will respond the same. And things that we've tried in the past, like nitric oxide and others, there's some people that respond very well, some don't respond at all. And as Steve mentioned, this is quite exciting that some ways to predict which patients don't respond to DCA based on a single blood test prior to starting. So that's an important idea. Targeted and personalized treatment better. And that's the goal of this. It's in the estrogen pathway, and you all have learned this. It's still much more common in women, 2 to 3 times more common. And yet, you have volume hypertension as a man, it's worse disease than it is as a woman. So there's lots of potential roles of estrogen and testosterone, hormones, and volume hypertension biology. And so I don't know much about an test result. What can you mention yesterday? I don't know if you want to tell us a bit about being a woman versus being a man.
Well, I know about that. But about that, so I'm not very familiar with the experience with this drug, but I think I think we're still trying to figure out why that's important in pulmonary hypertension. Why is it that so many women get this disease, yet they do better than the men that get it? So if you're a man, you're less likely to get pulmonary hypertension, but you're more likely to do poorly when you get it. So it's really there is something very important there. We think there's some evidence that perhaps it's the way that the pathway metabolism and estrogens are metabolized, and could be byproducts of estrogen that are harmful and could be contributing. The other possibilities as well. So, the paradoxical thing is that estrogen otherwise is very protective for blood vessels. I mean, there's very few drugs or agents that are as protective as estrogen. The blood vessels love it. It makes them happy. It prevents damage. There was even a trial in cardiology of coating stents with estrogen to help repair the blood vessels that are damaged. So it's not clear, what that connection is actually. So we'll see that this trial will be important if there's a positive effect, then obviously we're on the right track. But clearly, if this doesn't work, maybe we've just got the wrong idea, and we've got to look at it.
Does that just block estrogen? Is that how
It blocks the enzyme. It's
it works?
it blocks an enzyme that metabolizes estrogen.
So actually, it's stem the lifespan of estrogen.
Yeah, well, it prevents, I think, again, I'm not an expert, I could be wrong, I think it prevents generation of a byproduct. And there's evidence that that may be contributing.
Super. Let's move on to the last section that I want to talk about. This is a very interesting idea. We talk about plugging of blood vessels, and we showed you some slides earlier today that show that. That's not all that happens in pulmonary blood vessels that are feeding the lungs in pulmonary hypertension. One of the things that's quite interesting is we have an incredible network of blood vessels, starting with the main artery, each lung, which is bigger than my thumb, and then it gets smaller and smaller and smaller down to microscopic vessels. I can't imagine you put all that together, miles and miles of blood vessels. And so patients with hypertension, doesn't show this in beautiful work in animals, they lose blood vessels. They have less blood vessels. This idea of degeneration or disappearance of vessels. And it seems to target that some cells in the blood vessel walls are dying. And they die of this, it's a body that disappears. And so this is what everybody wants to talk about, gene therapy. It's obviously a world-leading authority on gene therapy. There's many trials on this, and it's focused on pulmonary hypertension in the last few years. John published the first gene therapy trial about 3 years ago now, a small series showing that it could be safely done. And we will just have them summarize what that first study facet told us about gene therapy, especially cell-based.
Oh, me? Okay. Well, actually, the brainchild's sitting beside me here, so. Oh, okay. Well, that's very charitable, but I got to say, Duncan keeps on pointing to me and really giving me the credit for doing any of this, but it's all Duncan. So basically, infusion of cells, which actually are harvested from people, so the patients get their own cells as opposed to somebody else's cells, which might stimulate your immune system to attack them. They're your own cells, so you live harmoniously with them, which is kind of cool. You can actually train them, if you will, to grow into blood vessel cells. You can identify them and grow them into blood vessel cells. And these sort of immature cells, you can actually get and insert particular genes or enzymes to produce a protein or an enzyme or a compound. And in this, Duncan was able to cleverly inject a gene which produced nitric oxide, and you know the importance of nitric oxide. So you could actually use these cells as a messenger to deliver nitric oxide into the body. So you can inject it into the lungs, and then they would naturally go into the lungs and stick in the lungs. But not only are they delivering nitric oxide, but these cells in of themselves have some natural ability to kind of heal the lung, or like maybe even grow new blood vessels, but certainly improve and potentially reverse some of the changes that have been described so far. And so this was really Duncan's early work in animals, and this was the first trial, not the first trial, there was a Chinese trial before that which was published showing some improvement, but this I think was perhaps a more well thought out trial and a more strict trial in how the cells were developed and the type of cells and the reporting. And what we were able to demonstrate, I mean the larger we, which was myself, Duncan and his team, and also David Langleman in Montreal, was that when you inject this into people over a period of 3 days, and one, it was safe, didn't block the circulation more, so nobody got sicker, because you'd worry about like sending cells into the blood vessels that would cause more pulmonary hypertension. That didn't seem to happen. And in fact, when you look at the pressures in the lungs, initially, quite surprisingly, there seemed to be an improvement, which I don't think was entirely expected early on. Like it was over the period of the 3 days of observations that the pressures would actually start to go down and the heart function would slightly improve, which I think was kind of unexpected. And the even more unexpected thing, and something that the earlier trial demonstrated from China, was that effect seemed to be a bit lasting. So it led to maybe some downstream improvement over a period of time, suggesting that that effect may be sustainable or continue. Those cells that are stuck in there continue to do something positive, and the enzyme continues to produce nitric oxide, which might have a not only lasting effect but an ongoing benefit. So I think that has led to Duncan's lifelong work in blood, sweat, and tears to develop a larger trial called Sapphire. And so maybe I'll hand it over to him. Was that an adequate summary?
Yeah, pretty good. The Chinese drug trial did not have phenol transfected cells, so that's something that is quite different from what has been done before. And our studies in animals suggested that really the gene part of the therapy is really important. And so if the cells are beneficial for it, sort of synergistic effect. So that's what we're thinking anyway. So yeah, John, of course, was quite precise in what he said. So we did have some signals, I guess, in a very small trial. We were very careful interpreting that because there's no control group. So we can't say for sure that it was working, but certainly it looked like it was well tolerated, and the direction was positive. So we're very keen to do this in a way that we can really get an answer and to know to what extent this may or may not be helpful for our patients. And because it is the patient's own cells, as John pointed out, we can give this repeat. And when you think about pulmonary hypertension as a chronic disease, it's hard to imagine if one delivery of cells models those would have enough to give you a long-term benefit. But the effect should be cumulative. So if we give repeated dosing over a period of time, many people get additive effects that could be much more durable. So that's really what the Sapphire is doing, so it's a repeat dosing. So every patient that is enrolled in the study will get at least 4 doses at monthly intervals. And then we'll see what kind of effect we have. And there are 3 arms, so one arm will actually get 8 doses, so we'll repeat. The second 6 months we'll repeat the dosing. Again, so what we'll learn, I think, is this actually effective at 6 months? So we'll have a comparison between patients that receive cells or placebo at 6 months, and then there'll be a crossover. So in that crossover, we'll be able to know those that got cells, and then we'll give them placebo the next 6 months. How durable is the effect? So does it last for the next 6 months? And that's important to know. And we'll also look at that third arm is going to get the double course of cells. We'll know whether we get further benefits if we give them. So I think this trial got thoughts back to the design and the idea that to learn as much as possible about this, and also to take it to a point where we've really done our best with the technology we have to give the adequate amounts of cells and give the opportunity for benefit. Obviously, if the trial isn't showing benefit, then we'll know this isn't something that should persist.
So, can I just add something? Duncan's very modest, and I think we owe him as a group, and hopefully you as patients, to his perseverance. Like, you think scientists are smart? Successful scientists are less smart than they're stubborn, and they persevere. And Duncan, honest to God, he must be the most pig-headed person in the world, because he has pushed beyond belief in getting this done. Like, he has gone through so many obstacles within his own institution, through review boards, through regulatory agencies, through manufacturing hurdles, through trying to get money. And I just think we all owe him a debt of gratitude. So I just wanted to shout him out and give him a hand.
Thank you, John. And I have to say, after a long journey and many hurdles, we actually found out, so we got our first site, which is now communication. So, and we're hoping to roll it out across Canada over the next 6 months or so. So it's been a long time coming.
Very exciting, wonderful. All right, so last slide has 3 question marks up there because there are some other new exciting possibilities. I want to sort of see, because again, I mentioned their lab, besides doing applications, will also study their own cell human mice, and they take cells from humans and study these cells to understand what's happening in each cell that makes that normal. It's very exciting ideas, including a clinical trial, something that's tailored to basic science research. So Steve, if you would enlighten us on that, please.
Yeah, very briefly, as we just discussed, pH is a complex disease. Part of it is going into the fact that cells within the blood vessels do proliferate and leading to progressive occlusion of the vessels. This is part of the mechanism. There are other mechanisms like vessels or the loss of blood vessels. And so the vessels that proliferate look like very much like cancer cells. So those are not cancer cells. So pH is not cancer, but they behave like cancer cells. And one of the mechanisms why they are able to behave is because they are able to repair themselves very, very fastly and effectively, pretty much like cancer cells. And we have access to medication that blocks the capacity of those cells to repair themselves quite selectively. So that's cancer cells or cells that are within the blood vessels of the lungs of pH patients, and it blocks the capacity of those cells to repair. So this is self-proliferating. So we very recently proposed a clinical trial to assess the efficacy of the drug that has been used and approved for the treatment of cancer. This is a drug that is taken by pill with very, very few side effects as compared to the traditional chemotherapy. And we proposed to assess that drug in pH. So at this stage, we we know we have access to the medication, and we just apply to get funds to manage all the clinical trial, and we get that money to do money.
Does that medication have a name or just a number?
It has a name. It's sort of approved by the Health Canada. It's called Moderator, and so this is the drug that is currently used in Canada for another disease.
Very exciting, wonderful. So I hope that gives everybody a sense, not only the tremendous progress that's been made over 20 years, to extend effective medications, but also very excited about new molecules and new pathways and new medications that are at the point to be giving us something more to use. I'm going to challenge my panelists quickly just top of your head. Most effective therapy that we're going to have in the next 5 years from this to talk about. None?
What two of them are going to trials?
Well, it is working.
Combination between these. No, it's going to. I think I'm voting. Well, I'm biased. I think this stem cell stuff is very exciting. So I'm going to. Sorry, Steve. I'm going to try for the stem cell.
Stem cells. But okay, you said stem cells. You can't say the same thing. So the other place to talk about.
Maybe what I will say is, what we're doing now is just touching the surface of what this technology can do. So if there's any positive signal in this trial, I think the technology is now being developed for cancer immunotherapy. So to genetically modify inflammatory cells to go after cancers. There's a whole bunch of companies developing all kinds of technologies that give us tremendous ability to engineer cells, to control when you turn on and turn off the gene. So these technologies which are now being developed, we can use them if it looks like stem cells and gene therapy working before I mentioned. And in the next 10 years, we can develop really, much more effective approaches. So this is the beginning. We're doing hope is good enough, but boy, there's a lot of potential for doing better. So we'll answer that one.
I don't think I have a question, but that's okay.
See, I need to be more politically correct, John. I say that maybe that PH is a complex disease and a very heterogeneous disease. And I think it is very unlikely that all patients respond to every therapy. And I think we've been talking about many new drugs. Many of those drugs may have potential side effects as compared to the drugs that we use currently. We've been talking about immunosuppressive drugs. So I think it's going to be a challenge for us to try to target the appropriate patient that will that there is more likely to benefit of that drug, and that would make the risk benefit ratio acceptable to that patient. So I believe stem cells is exciting, and targeting the proliferation of the cells that are within the bone arteries is also interesting. But even with the drug that I just talked about, I think it is unlikely that everybody will respond to that.
Wonderful. Please join me all in thanking our three wonderful panelists. So with that, we're coming to the end of my hope for it was fantastically. I know I had one other day. I hope you got a sense of the tremendous progress, the excitement that we all see, not just today, but going forward in the future, the optimism that we even more than all we have been able to. And the most important of the back to you, if you don't do this with your help, we'll invite you to connect with us, engage with us, push us, tell us what we're not doing a good job of, and tell us what you need us to do more for you. So a very great big thank you to all of you for joining us, and we'll see you the next time.